Hello, my name is Erin Newburn and I lead our field application scientist team at Personalis. Today I'll be sharing some details with you on our platform, ImmunoID Next, which is an enhanced exome and transcriptome platform used for composite biomarker discovery for precision oncology. I'll be walking through the assay design, and I'll also present a recent case study from a cohort of metastatic melanoma patients that were treated with immune checkpoint blockade, where we integrated several different molecular features from our platform to help better define the responders and non-responder patients. So first, a bit about our company at a high level. We are a provider of advanced genomic sequencing and analytic solutions to support the development of both personalized cancer vaccines as well as for translational research in immuno-oncology biomarker discovery. Our patented ACE technology, or accuracy content enhanced technology, it forms the foundation of many of our oncology products that we offer. And we have a company mission of really providing the most accurate and comprehensive data to help advance cancer treatment therapies. Personalis was founded back in 2011 as a Stanford University spinout. And our headquarters that houses our CLIA-certified CAP-accredited laboratory is located in Menlo Park, California. And we've highly invested in the operational infrastructure and sequencing instrumentation in our lab. And we are now one of the largest clinical sequencing labs in the US. And we have a very large and growing customer base. So as we've seen, the approaches to the treatment of cancer have evolved immensely over the last few decades. And what we've witnessed is this migration from a one-size-fits-all approach with regards to chemotherapy and radiation to the adoption of a more targeted, biologically informed modalities. And despite some of the very promising results we're starting to see in patients that are treated with various immunotherapies, with the advancement of technologies such as NGS, it's clear that further improvements in the treatment of cancer will really be driven by the generation of more patient and tumor-specific molecular insights. And as we continue to take on the various obstacles of immunotherapy biomarker discovery, we also know that these are very precious, critical patient samples that have been acquired. And traditional processes have often required multiple samples to be used on multiple technologies to get the essential data needed in a study. So for example, perhaps sending a blood sample out for HLA testing, another tissue section to look at the mutational landscape coming from the tumor, maybe another tissue section to develop expression signatures. And the result is that sometimes investigators have difficult decisions. There maybe are trade-offs about which particular assay can be ran when there is limited sample, or also dealing with complex data integration when you're working with several different platforms. And so ImmunoID Next was really purpose-built for immuno-oncology biomarker discovery and designed as a solution to use a single sample preparation and to consolidate multiple biomarker assays into one. And so we designed our approach, ImmunoID Next. Our vision is that this can really be a one-stop solution to elucidate very comprehensive biology, maximize data generated using this single tumor and normal preparation. And this is also one of the first solutions to enable a very complete characterization of both the tumor and the immune-related components of the tumor microenvironment. So as you'll hear about in the talk, we provide investigation into critical areas such as detecting neoantigens, HLA typing, digging into tumor escape mechanisms, oncoviral detection, the ability to look at the TCR dynamics. And again, this is all coming in a sample-sparing way. And so this platform, our platform, is really validated for the today's clinical biomarkers, but it also will enable the discovery of next generation markers and signatures. So in our lab, we receive a variety of different starting sample materials, FFPE, whole blood, fresh frozen, PBMCs, although the majority of the samples that we receive are FFPE from our customers. And on the left, this is a snapshot here of some recent FFPE materials that have entered our lab. And as you can see shown here, they come in a variety of quantities and qualities. And this is frankly the reality of working with clinical FFP materials. However, we've optimized to work with this more challenging sample starting material. We do a dual extraction of both DNA and RNA, and we can work with as few as 4 slides. We will assess both the quantity and quality from those materials. On the right, this is demonstrating a cohort of samples recently that our lab has received with various sample formats. You can see that regardless of whether we started with a fresh frozen tumor, a more archival FFP material, or a freshly prepared FFP material, we have the majority or a large percentage of the samples are passing both our DNA and RNA extraction QC criteria. So again, we've optimized to work with more difficult sample starting materials in our process. The ImmunoID Next assay itself is really powered by a very innovative assay design. First, by the depth of sequencing coverage that we provide, as well as the augmented proprietary ACE technology that results in enhanced sensitivity and specificity. From our tumor specimens, we run our enhanced exome, which consists of 300x mean coverage across the entire footprint. However, this utilizes, again, our proprietary ACE approach to provide augmented coverage in difficult to sequence genomic regions across the entire footprint. And I'll speak a bit more about this on the next slide. But what results is much more uniform coverage across all 20,000 genes as compared to a conventional exome. We pair that with our enhanced whole transcriptome sequencing of the tumor, delivering 200 million total reads. But secondly, we also provide boosts of ultra-deep coverage in critical areas of importance for oncology. The first being we provide a boost of coverage, clinical grade sequencing coverage on a set of cancer driver genes, 248 cancer driver genes associated with both solid tumors and hematological malignancies that we boost to 1,000x coverage, which gives us the ability to look at very low abundance mutations, but also, as you'll hear about, does also enable diagnostic capabilities from this platform as well. We also boost and enhance the TCR and BCR genes, which allows a look at the immune repertoire coming from these specimens. And also within this supplemented design, some other additional benefits and areas that we've targeted include the HLA genes, MSI-related loci, as well as oncoviral genes. And so as you can see, this really makes this assay kind of a first-of-its-kind approach. It's been very carefully designed, especially to allow a characterization of the immune-related components as well as diagnostic capabilities. And so ACE, our accuracy content enhanced technology, forms the technological foundation of many of the assays that we perform at Personalis. And the problem with kind of the standard exome and transcriptomes out there are that they have sequencing gaps in coverage and inconsistencies. And this is a nice visual of that shown here, where if you're looking at a particular gene, what we've done here is concatenated all the exons across the gene, and you're looking at gene-wide coverage using a typical exome approach, and you have what I like to refer to as peaks and valleys across the gene. Area is very sufficiently covered, but other areas that are more problematic or difficult to capture, where you may be completely devoid of coverage or have suboptimal coverage. And this is what we've recognized and addressed with our proprietary ACE chemistry. And our goal is to achieve more uniform sequencing coverage gene-wide. So this demonstrates the data that our customers would receive across this particular gene where we've enhanced. So they would receive basically the blue plus green, where we've enhanced and filled in some of these gaps to give more uniform coverage throughout the footprint. Also, accurate transcriptome sequencing is also critical as you're defining your genomic solutions for biomarker discovery. And we apply our same ACE technology to our transcriptome approach. And in many of these cancer studies, FFP samples are the materials, of course, that are available for analysis. And in the past, these samples have been quite challenging for genomic, but especially transcriptomic work. And so typical enrichment methods for RNA sequencing like polyase selection or ribodepletion have known limitations, such as just the inconsistent removal of ribosomal RNA, or also just the inability to work with more degraded materials. But methods such as ours, which is an exome capture enrichment protocol with our ACE cancer transcriptome, it allows you to overcome these limitations to work with a more degraded sample or more limited materials. And so in our approach, we use our optimized ACE probes targeting the exonic regions with our optimized chemistry to enrich for the messenger RNA. And so just as I demonstrated from exome data on the previous slide, this is showing a representative transcriptome sequencing gene results where we have both the blue plus the green to result in more uniform coverage across the footprint. And so in biomarker discovery, it's also critical to have an understanding of the sensitivity and specificity of variant detection in the assay you're selecting. And so we have performed a strong analytical validation of our ImmunoID Next DNA and RNA sequencing assays. And so I'd like to share a few slides with you on our approach. So in our approach, we procured tumor-derived cell lines and corresponding match normals for validation of small variants in both DNA and RNA. So here we're looking at SNVs and indels of 1 to 50 base pairs in length. And so we extracted nucleic acids from these samples, created libraries using our ACE preparation protocol, and sequenced on Illumina NovaSeq at a 2 by 150 base pair protocol. And so we then ran our somatic DNA and RNA variant pipeline, and then analyzed the detected variants according to a gold set of variants. And so we define this gold set as variants that were corroborated by at least 2 independent sources, so published in the Cancer Cell Line Encyclopedia from the Broad Institute's database and procured from ATCC. And so this represented a set of over 1,300 SNVs and 42 indels. We also looked at limits of detection by simulating tumor purity and generated dilutions of the cell lines by mixing the tumor-derived cell line with the match normal to represent purities of 5 to 80%. And so from this approach, I'm pleased to share our results. First, here on this slide from the Exome, we had very high sensitivity and specificity for not just SNVs, but also indels, with an overall sensitivity of 99% for SNVs and 98% for indels. But also in the table here below, you can see our performance at both a 5 and 10% minor allele frequency. Again, high sensitivity and specificity from our exome. Likewise, from our ImmunoID Next transcriptome, where again we're delivering 200 million total reads, we also have high sensitivity for small variant detection with 97% overall and 100% sensitivity for fusion detection. So taken together from both the exome and transcriptome results, this demonstrates the sensitivity and specificity of these assays, and at a limit of detection as low as 5%, as we've shown, this is more in line with the performance you would expect from a targeted panel, but here we are just demonstrating this performance from a broad genomic footprint. So additionally, another common challenge in biomarker discovery is taking on the challenge of analyzing the data that's generated from whole exome and transcriptome solutions. And so we've created various analytical modules to allow our customers to generate, informative immuno-oncology insights from the very comprehensive DNA and RNA data delivered with ImmunoID Next. And these algorithms evaluate the broader cancer ecosystem, and many of them are highlighted here. Our neoantigen ID, which includes NGS-based HLA typing, our repertoire ID, our immunogenomics ID, our microsatellite instability reporting. And basically the assay and the informatics have been really co-developed within our design. But to really illustrate to you how both the assay and the informatics can be put to use, I'd like to walk through a recent case study with you that was performed by our Personalis R&D team in collaboration with Dr. Sekwon Jang's group at the Innovashar Cancer Institute. So for this study, we've so far taken a look at the first 52 patients, and these patients all had late-stage unresectable melanoma. They were treated at the Innovashar Institute with an anti-PD-1 therapy, either pembrolizumab or nivolumab. They were evaluated according to RECIST criteria. We received the pre-treatment FFP tumor blocks and the corresponding matched normal blood sample. We ran our broad immunogenomic profiling assay of ImmunoID Next on these specimen, and then we're able to take the molecular data in conjunction with the clinical response information to look at biomarkers, but also to dig into potential tumor resistance mechanisms. And what we wanted to look at first was the mutational landscape of the tumor specimen in this cohort. So in our melanoma cohort, we found when we looked at tumor mutational burden, this was not associated with overall response. And also when we stratified by either high or low TMB, it was not associated with progression-free survival. However, from our neoantigen ID analytical module, when we looked at the neoantigen burden from the tumors, we found that it was associated with both overall response as well as with progression-free survival. So the next thing we wanted to do was to look a bit further in our cohort, especially some of these outliers that are highlighted here in the various colors, these individuals that had a higher neoantigen burden but did not respond to treatment or only partially responded to treatment, to try to determine if there might be other resistance mechanisms that might be at work here. And so we first evaluated this partial responder, again, a partial responder, but had higher neoantigen burden and did not respond here to anti-PD-1. And we looked further into the antigen presentation deficiency defects, such as somatic loss of heterozygosity. So from our HLA-LOH analysis, we found in this patient that there was a chromosomal deletion that resulted in the removal of one allele in all three of the A, B, and C genes. And so this is potentially detrimental to the number of neoantigens that can be presented for immune surveillance in this particular individual, and may explain why they only partially responded to anti-PD-1 therapy. We then looked at another non-responder. So for this patient highlighted here, where again we observed a higher neoantigen burden, yet the patient did not respond to anti-PD-1 therapy. And in this patient, we detected a somatic mutations in two HLA class 1 genes, HLA-A0201, HLA-B1501. One was a stop gain mutation, the other was a splice site mutation, likely leading here to the loss of surface expression of these proteins. In addition, when we looked at the full list of predicted neoantigens from this patient, we found that almost 40% of the predicted neoantigens were likely to bind to these two mutated alleles. Again, this is potentially suggestive of may play a pivotal role here in why this patient did not respond to anti-PD-1 therapy. And then we looked further at some of the other non-responder patients. And for this particular non-responder patient, we also looked at the genetic variation in the antigen presentation machinery. And what we discovered in the analysis is we observed a somatic frameshift variant within beta-2 microglobulin. And of course, B2M is a key protein involved in the formation of HLA class 1 molecules. And this frameshift variant potentially limits the correct HLA class folding and antigen presentation. And this is consistent with other studies in the literature that have showed similar mechanisms of resistance. So as you can see so far with our approach, we can report out on tumor mutational burden, neoantigen burden, but we can also integrate key molecular features to create more composite biomarkers and composite scores. And as we have seen recently from the literature, such as groups such as those published recently from Johns Hopkins University or Dana-Farber Institute, improved biomarkers predicting immunotherapy outcomes will likely need to be more integrated and multidimensional to really capture the crosstalk between the tumor and the immune system. And so the data highlighted here next shows, again, the power of having a high content platform such as a whole exome and a transcriptome approach, where we're able to create composite biomarkers and really integrate key features. And so when you're doing a tumor mutational burden assessment, you're basically counting up non-synonymous somatic mutations. And also, even with neoepitope or neoantigen burden assessment, you're basically counting up neoepitopes. And this is a first step, yet if these neoantigens are not able to be properly presented to the immune system for surveillance, how relevant are they in the assessment of the mutational landscape? And so what we saw was when we took into account the HLA mutational status of the patients, so basically looking at HLA loss of heterozygosity and somatic mutations in HLA and B2M, and removed those neoantigens that were predicted to bind to those mutated alleles, we found that creating this adjusted neoantigen burden yielded it further significance when we looked at responders and non-responders. So this adjusted neoantigen burden was able to better parse out responders and non-responders in our melanoma cohort. Also from our case study, we wanted to look at the TCR dynamics, and we observed significantly higher clonality in responders when compared to the non-responder population. And remember, these were pre-treatment FFP tumor blocks, and so this data is suggesting that higher pre-treatment clonality may be predictive of patient response to anti-PD-1 therapy. And so to summarize from the results here from the case study, from this look here at the first 52 patients in this metastatic melanoma cohort, what we were able to see is that when we looked at neoantigen load, it was a better correlate with overall response and progression-free survival than TMB. We were able to look into some of the potential tumor escape mechanisms that might be at play. And also, as shown here in our data, this adjusted neoantigen burden, this is a nice example of how our analytics can be integrated to create a more composite score. So here in our cohort, we took into account neoantigen burden as well as mechanisms of resistance, and that was able to give us a more statistically significant separation in responders versus non-responders. And we also looked at the immune repertoire and found TCR beta clonality correlated with response. So hopefully what I've shown or demonstrated here in this case study is how ImmunoID NeXT can be used to very comprehensively characterize both the tumor and the immune-related components of the tumor microenvironment. And we can do this in a very sample-sparing way from FFPE materials, and we work with just as little as 4 slides. And so to summarize the assay features and the design of ImmunoID NeXT, here's a nice table to quickly outline the features that are delivered in this design. As mentioned, this assay includes optimized uniform 300x whole exome sequencing coverage of the tumor. We also pair that with deep sequencing of the matched normal, 150x normal exome coverage. We also employ transcriptome sequencing of the tumor, and we deliver 200 million total reads. We also enhanced the TCR regions as well. We can run this assay in a tumor normal configuration. It's preferred as it does, of course, give more specificity for somatic analysis, but we can offer this in a tumor only configuration as well. And we boosted sequencing coverage in many key regions like cancer driver genes, TCR genes, as well as HLA and MSI loci. And further, we are also targeting the oncoviruses and can detect the 7 most common oncovirus types. And again, we do this in a very sample-sparing way. And so in addition to innovating with our ACE platform and our ACE design, we've also built an enterprise-scale genomics management system called Symphony that orchestrates the many steps in the process and tracks samples from receipt through extraction, sequencing, and analytics to data delivery. And this system allows us to lock down pipeline versions and content for the duration of a clinical trial or a given project. It also enables service in an expedited fashion following both RUO or GCLP guidelines, depending on the project need. It also allows us to be very flexible in data format reporting and in meeting different data transfer specifications that are unique to each pharma clinical database. And so when we engage in a study, our customers are assigned a dedicated project manager that can be that singular point of contact throughout the project. And our Symphony system allows our project managers to easily provide real-time updates to our customers on sample status. Further, of course, there are other critical components that are needed in meeting the operational infrastructure needs and biomarker discovery, and I've listed some of these considerations here. Having the instrumentation, the automated processes to meet fast turnaround times, and at Personalis, we have significantly scaled our sequencing operations with the acquisition of the NovaSeq. We've also invested in lab automation and robotics. And other investments needed include the computational power needed for the various analytics that we just described, as well as having workflows, quality management systems, and the regulatory support through the validation of our assays and informatics. And this all places Personalis in a unique position to support the transformational immunotherapies that are being explored today. And in my last few slides, I'd like to just mention some additional capabilities that we offer at Personalis. So we have various assay options and configurations. Our latest ImmunoID Next with the various benefits and enhancements that we've just discussed, as well as our ACE ImmunoID, our previous platform built on the same proprietary technology, but here again delivering at a different level of coverage to meet certain research needs. But we also have some additional sequencing solutions. We have a comprehensive cancer panel. We have whole genome sequencing offerings for both cancer as well as non-cancer applications. We have different enrichment strategies for whole transcriptome sequencing, such as poly-A and ribodepletion, along with our ACE enrichment that we just discussed. And also the ability to deliver pathology when needed, H&E staining, PDL staining, and also microdissection. And another exciting update from Personalis is at the end of 2011, we did launch our clinical NextDx test to help oncologists identify therapies and potential clinical trials for cancer patients. The Personalis NextDx test, it includes advanced analytics to provide a diagnostic report on genomic alterations in medically important cancer genes. And shown here, this is a screenshot of this diagnostic report out. So we do report on MSI status as well as emerging immunotherapy biomarkers such as TMB are included in our clinical report. But here it's important to note that our TMB is exome scale. And as opposed to a panel-based assessment where this is bioinformatically extrapolated, we give a true exome-wide assessment of TMB in our report. NextDx then, this report is coming from the boost in coverage I mentioned at the start that we provide for the 248 cancer driver genes, and boosted very deeply here in coverage with 1,000x coverage across this critical clinical content. Currently, NextDx, this test is supporting academic key opinion leaders and pharma clinical studies. And for us, this is really exciting because it represents the first step towards a companion diagnostic. And so we really are a partner throughout the development process, throughout drug discovery. And currently we are supporting many pharma and biopharma customers in their clinical development and translational research of their phase 1 through 3 trials. We now have clinical testing support with the launch of NextDx LDT. And again, this represents our first step towards a companion diagnostic. And our BD team is now working on a path, and we have a strategy for our CDX. However, we also, as mentioned, offer general research solutions for both discovery and preclinical work. So in my last slide, just to summarize, ImmunoID Next is designed to be a universal genomics platform for immuno-oncology. Next combines highly sensitive exome and transcriptome sequencing with advanced analytics and best-in-class delivery, and with the needed scalability. This assay is performed in our CLIA and CAP laboratory on NovaSeq instrumentation, and with the reliable support of both our project management and also field application scientist teams. And so we are very excited with these extensive capabilities to provide a complete look at the cancer ecosystem and also enable consolidation of multiple biomarker assays into one. Thank you very much for attending this virtual presentation. Hopefully this gave you some insight into our solutions and applications for tumor immunogenomics and biomarker discovery. Thank you.